just before the lecture in this channel. And this list will continue, and all these links or posted message are some additional information if you want to go deeper. There are YouTube links, there are blogs, there are Twitter links. People discuss the topics around the VIBS concept we discussed about. So this week, we are going to go to some very basic concept in, yeah.
Sorry to interrupt. I just wanted to ask, could you please increase the font size if you can for the screen?
Yes, so this is not the PowerPoint yet.
Okay.
Yeah, so this is the first semester we use this big room, and I'm happy we have changed this screen to a larger one. It was small, much smaller than this. And I hope everybody is fine with this. Later, can you, so this should be okay, right? Yeah. So this is a basic concept in lineage algebra. We will discuss vectors and matrices. Those are the important. We will see what's the operations you can applied on to vectors and matrices. And then the second half of the lecture today will move into Cell Ranger. So Cell Ranger is a pipeline provided by 10x Genomics to allow you to convert FASTQ row sequence data you sequenced from individual cells using their droplet barcoding system. And then how can you use this pipeline to convert row sequence FASTQ into expression matrix? Remember, in the center of our analysis is this expression matrix. I always use a big X to represent as a variable. And this is a gene by cell, and some software package uses cell by gene. So that's just transposition of the matrix. It's okay. But this lecture we will solve the problem. How can you get from a FASTQ, your sequence from Illumina sequence files, and through this pipeline, you get this matrix. Okay, this matrix is the center of our downstream analysis. We will focus on, of course, the downstream analysis on this matrix later, but today we have to understand how this 10x genomic system works and how individual mRNA molecule can be read, can be counted, and also you can separate individual cells. Last week we discussed this gel bead system, and you have a droplet in the center, you have a gel bead covered with a barcoding, 10x genomic barcoding. But still, in each droplet, you have millions of copies of mRNAs, and how can you tell which gene this mRNA come from? So we solve this problem today, okay? So we will jump into vector and matrix definitions, then we will go through matrix multiplication, geometric interpretation of matrix vector multiplication. And we will mention about what is eigenvalues, eigenvectors. So I hope this lecture will help biology students bridge yourself towards the data scientist field, because when data scientists talk about the eigenvalue of eigenvectors, they won't explain that to you because assume you know what they are and why you are doing this multiplication and what that means. So we will solve it today, use very insightful examples. Okay? Yes?
So when we get the data, the raw data is only one file? It's just gene? Because I think from last week, we got 3 files. We have matrix file, and the barcode file, and the gene.
Yeah.
Right? 3 files.
Yeah, 3 files. 2 files are the labels of the gene and labels of cells. So that's just extra information. But in the center is this big X matrix.
So
Matrix is
matrix we got from the raw data, or
yeah. So matrix. So you sequence. You put your samples for one sample, but there are many cells. That's cells distorted. So you go through this 10x genomics machine, CRIM controller. Then you get libraries for sequencing. Then you send it to Illumina sequencer, and you get FASTQ file. FASTQ files just short fragments. So you have to send these FASTQ files to 10x genomics cell ranger pipeline in order to get 3 files. And one is the MTX file. This is a matrix, the expression matrix file. Another will be TSV, this is gene list, gene name. And then another TXT, the barcode for the cells to tell which cell the barcode is labeled. So, but recently, Cell Ranger upgraded to another version. So 3 files have been combined into .h5 file. So you can have just one output file. This contains information about the matrix, the big X matrix, and the name of genes, the label of the cells. That's all.
So nowadays we only get 1 file.
They have options. You can have 3 files or 1 file. It's the same information. Yeah. Yeah, it's good to have this big concept on your mind, and so you don't lose. So data analysis will start from here. So we build representation of the information from the output files into computer. In the computer, you have a big X, that's gene by cells. Okay, gene 1, gene 2, cell 1, cell 2. And all the downstream analysis, we will play with this. So this will be bioinformatics analysis. So that's the center of our data structure. Okay, so this is directly bring us to why we are going to study matrix, because we play with data points. When we talk about data, so you can think we play with cells, so that will be the columns. And sometimes you want to play with genes. In that case, your data is by rows. Okay, each gene, gene 1 have a expression profile across this population of cells, and using this number to define the gene. But most of the time, we deal with the cells. That's by columns. Each column is a data point, and each a cell. And the state of the cell defined by the list of genes expression value. Okay, that's a vector. So to deal with this, we need a lineage algebra, and it's so essential for machine learning, because machine learning it's all about to understand the data structure, to be able to separate them, to cluster them. And if you have a label, you will know, okay, which label, the data points linked with this label have what kind of features. So everything we are talking about the data points can be studied using linear algebra. So this is, machine learning itself basically is linear algebra. Give you an example for applications. On your left, you have some data points, okay, blue points. So this is a 2D data set. Now, imagine yourself is a 1D animal. You don't see the overall picture of all this data points distribution. You can only sense, say, by your x-axis. So you move around the jungle of these data points. You go from this end to this end. You have some feeling how dispersed of this data are, all right? So you feel, okay, from here I move some distance, I touch the another boundary of this data cloud. So I have some feeling using my 1D sense. However, this is a 2D data, so your feeling of the structure is not complete. If you move from this end to that end, you will feel, okay, this data structure is so narrow, every data point seems clustered together, clumped together. So it really depends on which angle you use for your 1D measurement, okay? So this is a 2D example, but in reality, in the single cell data, each cell has been defined by thousands of genes, so it's a much higher dimension, but this is like an example. We can only use reduced dimension to understand the data structure, how they disperse, how far away from one cell to another, this is the boundary of this cloud, high dimensional. So what we can do with this, we use reduced dimension of a sense. So instead of starting with 2D, we use 1D, but we change our view of points. Instead of using this original x and y axis, we can rotate the data points, okay? So we change and rotate our x and y axis to allow our sense to explore this distribution of all the data points through either x or y or some angle which 45 degree between x and y and other degree until we found a particular angle which we feel we can sense the broad distribution of the data. So what that means is you found a new axis of x, and you can feel from this end of the data points to that end is maximized. All right?
Will that not affect the data points? Like they would change if the axis is being rotated? So
The data point itself not change. So you only change your observation. You change your x and y. Perspective. You can also think, okay, I don't change my x and y axis. I rotate the data. The shape of data is still in the same position. Everything changed. Okay. So you can also imagine you project or put a light and a shadow onto this plant, and then see how wide this shadow is. And you want to change the angle of the light until you reach the maximized shadow boundary. Okay, that shadow boundary helps you to understand how dispersed the data is, and for some angles is how narrow it is. So to give you the feeling. And also, if you have a two cluster of data, one is one cluster, another, this rotation of data points help you separate them later. Okay. Another practical application is like this. So you imagine this teapot is formed by all the data points and gives the shape of 3D. But this website say, okay, I only allow you to take one picture. Okay, so you have to select a good angle to take a picture of this teapot. to give the maximized information to the viewers if they have no idea of the shape of the teapot. So you don't want to take a picture from the top, from bottom, and from this angle, because the result of this 2D picture will not show you the most widely spread the points on the teapot here, 3D. Okay, so we are doing a rotation. Okay, you rotate your teapot or rotate your angle to take a picture in order to display the most featured data structure of this teapot to show people. All right. So all this operation we need to rotate the data point to display to solve this particular optimization problem quickly, analytically. So you can select any angle of this axis in order to achieve the result. You can try and a lot of different combinations and to get the most dispersed. So that's optimization, but we can solve it using linear algebra very quickly. There are a lot of resources. Certainly, you can get books, links, YouTubes, tutorials. But the purpose of this is try to, on the class, I try to solve your problem to save your time, okay? Basically, nowadays, every piece of knowledge you can learn online, there's somewhere in the corner, and you can find the resource. But if you solve the problem here, and save your time, okay? So allow you to share the language with the data scientists later, okay? So we're dealing the single numbers, so it's called a scalar. So if you have a number 3.5, that's only one number, that's called a scalar. The same language in MATLAB. And then if you have a vector, it's like a column vector. So this vector is 1 by n matrix, basically. It's just a list of numbers. The list of numbers gave you the property to define the property of this variable, A. So this bold A is defined by a list of numbers. So in our case, we are talking about the cells or genes. In more general case, in the machine learning, if you deal with real estate, this could be one house, and then there are how many bedrooms, or 3, how many baths, 2.5, and how many square feet, what's the market value. So you can have a list of numbers to describe a property. Or if it's a stock market company, you have a lot of other numbers to describe it. So the vector is the data we are talking about. So when data scientists talk about the data points and their data, they are talking about the vector. Okay. Most of this depends on the convention, and we use column vector as a data, okay, just like this. But in statistics, people use rows as a data. so it depends on which field we are talking about. So either by column or by rows, so you have to make sure you know what you are doing. By only look at the variable name, you only be able to tell. In quantum computing, people really smart. They invent this notation to tell, okay, this is a vector, a column vector, and if you do the other way around, this is a row vector. So it's called a cat and bra cat. But we're not using this. We're just use single variable name. And if you have more than one, okay, so this is like a matrix, and you use capital A, and in this case, it's a matrix of a 2 by 3, okay? So for the matrix, you can consider this is like a collection of vectors. So if we are talking about the column vectors, in this case, we have 1, 2, 3 columns. So this matrix is a combination, a collection of 3 data points, 3 vectors. And if you're talking about the rows, there are 2 data points, okay? Row 1, row 2, and defined by 3 variable or feature variables. Okay, as you know, we can plot vectors on this. So this is a 2D vector, so A, and then you have 2 numbers, x-axis and y-axis values, coordinates, and the vector always start from a origin 0, 0, okay? If it's 3D, you have a origin 0, 0, 0, and then you have 3 numbers to represent your data point in 3D. And the same thing for 4D. So almost everything you can expand from 2D to higher dimension. Almost all the rules, all the theorems will be applicable to higher dimension. So that's a 2D, that's 3D, you know. You can imagine your higher dimension. You have in this case, this vector have a capital, this n dimension, and then you have to think about, imagine. You can represent the values in this way. For example, there n equals 15. There's a 15th dimension of one data point. But it's useless because there's no way you can visualize greater than 3D. And in this plot, there are 2 vectors, A and B, okay? They all start from origin. Those are the common operations you can do for vectors. We can multiplicate vector by a single number, so it's called scalar multiplication. So if I have 2 numbers and you multiply it by 5, then you basically increase values 5 times to 5 times. And you can do addition, you can measure the length of the vector and their inner product, and we will go through those. So those are the major ones. Sometimes you say, okay, if I have a vector which not start from 0, not from origin, and what you can do is to move this vector back to here by, if you have a Q here, 6, 3, and you start your vector, instead of 0, 0, you start from 2, 2. What you can do is use 6 minus 2, 3 minus 2, and you get 4, 1. So this is a vector of the real representation of your vector, which is not start from origin. So if you have 2 vectors, you can do this addition and subtraction. There are geometric interpretation of those. So if you have A and a B, and your addition will give you a new vector, which is here, okay? So you draw these triangles. And the difference between A and B will be the link between them, but it's moved to the origin. So if you want to calculate the distance between point A and B, you can use A minus B and measure the length of this new vector, the results, okay? So we are doing this because we want to measure distance between 2 points. Or if you have a cloud of points, we want to measure the distance between your data point 1 to all others and data point 2 to all others. So you need to have a quick way to calculate the distance between data points. They are very useful and for your downstream analysis. So we need something like this, okay? So you need to know data point 1 vector minus data point 2 vector, and then measure the length of this result is their distance. Another concept is so-called inner product of 2 vectors. So in this case, the illustration is a 2D. So you have point A and B, and A have a length of x, B have a length of y. So this is vector. In this case, there are examples, small a and small b are 3D data points, 3 dimension. And the inner product of a and b is calculate like this, and you get 11. Okay, so this is very important, and we will discuss this equation later. Okay, you have vector x times y. Let me use cursor. We are not going to go deeper today, but we'll do this next week for measure the distance. Okay, so between 2 vectors. So now you need to know is inner product, or so-called dot product of 2 vectors, will be a scalar. So if you have 2 vectors, if you do dot product, at the end you will get a single number. Okay, so this is the inner product. The single number, this number has some meaning. So the meaning of this number is the length of x times length of the y, so and times the cosine of theta. So dot product has a meaning of this. All right? There are some links to help you to offline, and you can take a look of those. We don't have time today, but it will be important topic for next week. Okay, so we will have a cosine distance measured between 2 vectors. There are a lot of applications that will be center of next week's talk. Okay, so norm. And norm is the length of the vector, okay? In mathematical and data science literature, people put 2 bars around your vector, your data points, and that means the operation of take the first value in vector squared plus second value squared until the end, then take a square root. So if you have a data point like 3 to 5, you will get a square root of 38. That's the norm of vector. So the norm of vector is the length of this vector from origin to, that's how long this vector is. Frobenius norm is the same as this so-called this should be called L2 norm, and then basically the same. So you take each element in your vector or matrix, you do the same and sum it, and then take square root. So in terms of for vectors, Frobenius norm and L2 norm are the same. Another important concept is so-called unit circle. Okay, so we start from the center, the origin, and then found a point and draw a vector here, and make sure the norm of this vector is 1. And then we can find all this kind of vector form a circle, and this circle is so-called the unit circle. All the vectors or data points on this unit circle have a norm 1. Okay, so the length from this data point to the center is 1. It's very useful. That's as a rotator, so we don't care how long this vector is. We just found the direction, okay, for this. So this is like 2D, okay, this circle. If you think of sphere, you can have a 3D or higher. Okay, for the matrix, if you collect your data point, put in the matrix, then we have transposition, there are many other operations. And then you have matrix and the vector operations. Let's see. So first, transpose is easy to understand. You just rotate, and from this 2 by 3 becomes 3 by 2. Same here, if you have a gene by cell, and some software use cell by gene transposition, and you have this function in the Excel spreadsheet operation. Identity matrix is a square matrix by the diagonal elements are all ones. There are some addition operations you can apply. So if you have a matrix A plus B, it's the same as B plus A, or something like those. Scalar multiplication for the matrix is simple. So in this case, you multiply 2 to this matrix, and all the elements inside the matrix get doubled. And those are some of the properties if we are doing this scalar multiplication. Okay, so matrix multiplication will be the center of topic today we are discuss. And number 1, you need to know how to do it. Number 2, you need to know what that means. So why you are doing it. Do I have a erase? I need an eraser. Oh, yes, it's a good idea. So where I'm swipe this whiteboard, you maybe spend some time to read this story. So all the slides are with a yellow star is most important. And if you forget everything for today's lecture, just remember those. So I had one before. I don't remember which one, but this is the second one. Okay. Read about this sandwich making story, and then see. So online, there are a lot of material people teach, like data scientists teach other people how to do the matrix multiplication. They use wrong examples. They use like a square matrix multiplied by another. It doesn't give you the generalized concept of the match of the inner dimension. For example, we'd better use this as an example, okay, to do this matrix multiplication. Let's see this A. So I will have to to do A. So let's see A is here. So A is 4, 5, 3. So this matrix is 2 by 3 matrix. So A is 2 by 3. And the B, let's change the color. Three by 6. Remember, you have to have the same number from A to here. It has to be the same in order to do this multiplication. Otherwise, you just cannot do it. Okay? So this is called inner dimension. So you have two matrix A, 2 by 3, B, 3 by 2. So 3, 3 is in the between of A and B, so it's called inner dimension. The inner dimension has to be the same in order to do the multiplication. If you know the meaning of this is because you are working with the 3 type of sandwich. Okay, the inner dimension means 3 different type of sandwich. And the B is their gradients, the components to make this sandwich. So you have to give 3 recipes. Otherwise, you just cannot compute it. So 3 has to be in both A and B. So what do you have to do normally? We'll put this as 2 and 3 and put your 3 and 6 on the top. So I have 3 and 2 to 2. So I put this in a special way so I can know my results is in this junction site here, okay? So this is 2 by 3. This is 3 by 6. Let's fill that in, 4, 0, 0. Number 1, make sure the inner products match so you can do the operation. Number 2 is you put this arranged in this way because this gave you the sense what the output, the dimension should be. The output will be here. Okay, so what's the dimension of this corner? Is 2 by 6. All right, so this will be C will be the output is 2 by 6. So people use terrible examples, use a squared matrix to illustrate those general rules. That's really not good. So what's the product of this A and B? So what's the C? A multiplied by B. So A is a matrix, B is another matrix. They can be multiplicated because they have a matched inner dimension. So what's the result of this? So A is you and your roommate. You want to make 4 type 1 sandwich. You want to make 5 type 2 sandwich. And you want to make 3 type 3 sandwich. And these 3 different type sandwich have a different ingredients. And you have to buy butter, and that's what's a P. Bread and peanut. Yeah, jelly. And that's the amount, okay? So at the end, the product A and B gave you how many bread you have to buy, piece of bread, how much the peanut butter. In this case, you lost information of the 3 different types of sandwich. You only have this compounded or synthesized composite end product of the ingredients, the amount you want to buy. But you lost the information about what are you going to buy for this peanut butters and how you distribute it in the 3 different sandwiches and how much you will distribute them, okay? Then how you do this? You use each number here, multiply each of this, and take a sum and give the value here, okay? Questions?
So let's see the Y in the composite matrix, the A and B matrix. So it says 24. So this means that that is U. I mean Y is for you. So U requires 24 slices of bread.
Yeah, yeah, because you want to make 4. You want to make 4 sandwich type 1, so that's 8. Then you want to make 5, so you have to 10 here. And 3, 2, 6. Yeah, so at the end, in total, you have to buy 24 pieces of bread. Okay, so matrix multiplication put different gradients together in different ratio, okay? And the synthesizers that gave you the final product. Now I'm going to translate this bread and sandwich, different those into single cell data analysis language. Let's see, you yourself is 1 cell type, and your roommate is another cell type. And you want to be function, okay? You want to express some genes, then B, P, J, H, C, T are 6 different genes. Now, you, as a cell type, you express gene B, 24, value 24. That's you, and express gene, bread gene, B. And your roommate also express that gene as 24. And then you express peanut butter, 16. And your roommate express it, 12. Okay, that's your observation. So you observe this different cell type have a different gene expression. But this gene expression, like 24 and 16, this gene getting involved in different cellular process of biological functions. How many functions? In this case, we have 3 functions. That's 3 different types of sandwich. Okay, so this multiplication of matrix give you some different views of what you observed at the final product of this matrix. Okay, 2 cell types, each involved in 3 different cellular process. Each process require 6 different genes, have different combinations of the expression. Okay, just think about this concept. Then that's A times B is your observation. That's your expression matrix you get from a single cell. So we don't have observation of this matrix. We don't have observation of that matrix. We only have a C. So C is A and B. C is A times B. In our data analysis of single cell data, we actually want to do the opposite. We want to separate the C into A and B, so we know the biological process different cell type involved in and how much they involved in each, what's the proportion of that. Okay. So I move back 1 slide. So this just, how are you going to compute A times B give you C? Okay. So in this case, the example is 2 by 3 and 3 by 2 matrix. And then at the end, you get a 2 by 2 matrix. Remember, you put A here, B there, and if you have a 2 by 3, 3 by 2, at the end, you get a 2 by 2. Okay? That's how you compute the matrix. Questions? Again, this is most important, and you know how to do it, and you know some the idea behind doing this. Okay. The opposite to separate the product A and B become A and B is a decomposition. Okay.
So the A and B are from the same sample, just random matrix.
Yeah.
So 1 sample file, just.
You are talking about the single cell data.
Yeah, the A and B.
No, A and B is unobserved. No, we in single cell data analysis we don't observe it directly. We can infer. We can see. Okay, if I have A and B matrix, I know what the C should be. But in reality, we observe C. We don't know the A and B. Yeah.
Can you please go through in the composite matrix, the sandwich example, for A and B in the R column, in the R row, how, I'm slightly confused.
How this computed? Yeah. So in order to compute this 24, you look at this row against this column against this row. You put them together, multiplicate element by element, then sum together. Then you move to here, the second column, this row, and then column by column, and put together. Yeah.
In the Teams group, I put an example from the MATLAB website, and it's like color coded too. I don't know if it helps.
Yeah. Yeah. So this will be homework, just described. Okay. So you will know how to do it eventually. Yeah, I just want to make you think. If you have the end product, how can you decompose it? So this is a bigger question. And from here, so you're in out your dorm and you look at 2 students come out to buy those, you only be able to figure out how many sandwich student 1 and student 2 will make, how many type, unless you have extra information about, okay, they want to make 3 different kinds of sandwich, each of these, and that's why they buy, their shopping list looks like that, okay? There's a way you can move back, and there are numerical way to approximate the possible combinations. But you have to know the number of sandwich types. 3 is a number, so you have to know before you can do those, okay? Okay, so we have a general rule here. So if you have 2 by 3, 3 by 6, then you end up with a 2 by 6. Now, if I ask you to do a vector by matrix multiplication, in this case will be, for example, the 1 by 3, 3 by 6. The end product will be 1 by 6. Okay, the same rule, but just make this become a vector. So if you have a vector here, 1 by 3 at the end, the product of vector with, so this will be 1 by 6. Same thing, okay? Now, before jump to eigenvalue, eigenvector, I want to say, okay, if you have 2 vectors, you do multiplication, tell me what's the dimension of your product. For example, I have a vector A. This will be 1 by 5. And my vector B will be, have to be 5 by 1, okay? By 1, vector, okay? Still 1 by 5. Can we do multiplication between this A and B? We cannot, because this vector not follow the rule of those so how can we multiplicate these 2 vectors? Transpose one of them. So let's transpose B into B transposed. So now the dimension will be 5 to 1, right? So what's the result of A times B transposed? So the 2 vectors. So what's the dimension of C? 1 by 1. Okay. So this is a scalar, just a single number. So this is inner product or dot product. My question is, can we do A transpose times B? What's the dimension of C? Yeah, 5 by 5. So this is only 5 dimension. If you have millions or hundreds of, then this is really powerful. This is called outer product of these 2. So this is called inner, this is called outer. So if you, 2 vectors, if you do inner product somehow, and then you get a single value. However, if you transpose this, multiplicate that, and you get a huge matrix if you have a high dimension of the vector. So this is only 5. If you have a 5,000, then this simple operation will give you 5,000 by 5,000 value. Okay.
What's the criteria to select which one to transpose? Is it something like the bigger the matrix, the better it is, or something?
Yeah, depends on your application. There use of the inner product. Outer. We will see the examples later, okay? So inner product. So you don't have to remember those because you only need to remember how to do this, and you can derive or work out yourself inner product, outer product, what you can get. Just follow this rule, okay? I think we have to take a 5-minute break before we jump into this, the next topic. All right? Okay, now we are going to use an example to look at what is eigenvector and eigenvalues. So now you have to think a matrix like A. This is not only the collection of the data points. So before that, our introduction is this is a collection of the two data points. Okay, one column, second column. However, in this case, we will look at this as an operator. The operator means you apply this to a data point. We will apply this to a data point 1, 1 as an example. So what's the operation? Is we do multiplication. Okay, we multiplicate this matrix with vector 1, 1. So how can we write this? So remember we have 1, 1 here. So the answer, the results will be, so the matrix is 2 by 2, okay? And the data point is 2 by 1, 2 by 2, 2 by 1. So what's the answer of this? Should be 2, 1. Yeah, right? Yeah. So this times this times this, and plus, what's the results? The 3. This times 2, 6. Okay? So this is our multiplication of one squared matrix, 2 by 2 square matrix, times a data point, 2 by 1. And the result is 3, 6. It's another data point. So if you plot this out, the original is 1, 1, so it was here before the operation. This is our, so this is like a V, our vector. And this V before operation. The after operation, it becomes 3, 6. 3, 6 somewhere here. Okay? So that's A times V. So as an operator which apply to the data, what this operation of multiplication by square matrix A is to rotate the angle of this vector. Okay, rotate some angle and also stretch it, make it longer. So they changed the norm and also change the direction. So that's most of this operation will do onto data. However, there's a special data point. What is 510? Okay, 510 is a special vector. That's 1, 2. 1, 2 is special vector. So this is 1, 1 normal. 1, 1, 1, 2 is here. Let's call it a U. Okay. So this is a special vector, and the value is 1, 2. This special vector in terms of this operator, so they are a pair. It's not a special vector for every operator. This is a special vector for this specially defined A operator matrix. Why is it special? Because if you apply A onto this mu, you will not change the angle of the you so only make it longer. So if you do 1, 2, 2, 4 multiplied by 1, 2, you get 5, 10. The result will be here. Five, 10. Oh, should be somewhere longer here. Okay. So now what you can learn from this example is we will consider squared matrix not only as a collection of data points, but we consider it as an operator. This operator, once you apply this operator to other data points, normally it will change the other data points' direction and also the length. However, for this given operator matrix, there are special data point. But when you apply this operation on this data point, it only change the direction of this data point or this vector. It only stretch it or shrink the length of the vector. So for this particular example, I can write in this way. So this is x, okay? X instead of mu is x, okay? So operator A times x will not change the direction, but make it become 5 times longer. All right. Make it more generalized. So this is a constant. Okay. So if you write A applied to this vector, it becomes particular constant lambda times vector. So if you can find a relationship like this, now you found an eigenvalue. So this is called eigenvalue. X is called eigenvector. So you found a pair. You found a pair of vector and constant value. This pair is regarding to the matrix A. So you found eigenvalue and eigenvector for operator matrix A. So that's a definition of eigenvector, eigenvalue, and what that means. Then we will later expand this to a lot of applications using this relationship. I can tell you one relationship, the application, for example. And if you move this x come to here, there are operations for, so you can move, if you know A, and you know its eigenvalue and eigenvector, you can move this come to here, and it becomes transposed, okay? Some kind of transposition, transposed lambda. I have to check whether this makes sense or not, but anyway, if you rewrite this, you move x come back to here, just like 1 over n if you do the multiplication. But in this case, you have to transpose this or inverse that vector. Then you actually decompose a matrix A into a vector plus times a value. So through this operation, you learn the feature, you learn the relationship between data points inside matrix A. Remember, we have this sandwich you and your roommate buy and make a matrix. How can you decompose this into sum of the A and B multiplication? So in this case, you have A and you decompose it into some kind of vector and constant value multiplication. Okay? So this is just one of the application to use eigenvalue and eigenvector, why people always talking about those. So we have to move, and we will have more examples, not today, but for the following lectures to work on those. So PCA rely on this, okay? PCA, the A is called covariance matrix. Then you do this eigen decomposition to find loadings, okay? Okay, matrix inverse, and so if you have A, then you inverse it, and the multiplication of between them should be an identity matrix. Okay, it has to be a square matrix. And the trace, sometimes people use a trace that will be the sum of the diagonal elements. So I will skip this determinant. Again, so we have a start, and this a star in this particular slide. So I hope you can use your own language and try to explain what is eigenvector and what is eigenvalue. This A has to be squared matrix. If you have a non-squared matrix, you don't have an eigenvalue and an eigenvector. Okay. So this pairs of value and vector, sometimes you have to have more than one. Okay. In this case, we have only 1, 2, but you have another one on this space. So those pairs is a way to help you to understand square matrix, the feature within this squared matrix. We're not directly look at the values inside here, but we use the consequence of it used as an operator to understand the structure of the data inside a matrix. All right, sometimes people say write M, so it's the same thing, okay? So just the squared matrix, M times M, okay? Anything else? This is another example, okay? We have this, but that's another A matrix squared, and this is 2 eigenvector. eigenvalues, their corresponding eigenvectors, okay, there. And you know how to do this multiplication between one matrix with another or one matrix with another vector. So you can play with those numbers and try out on the paper to see, okay, this relationship will hold. Close your eye and remember the formula. It's not hard, but you have to understand what that means. So in those examples, we are using non-symmetric, but just a squared matrix. This is a symmetric matrix because the off-side values, off-diagonal values are the same. So if you do the transposition, and this will be the same as its original. So in that case, we call it symmetric matrix. And a squared symmetric matrix, mathematically we can prove that all the eigenvalues will be a real number. There's no complex number involved. Otherwise, there's no guarantee. Okay. And this is not a symmetric matrix because here is a 3 and that corner is a 2. They are not the same. So if you transpose it, you flip it, and you don't get the same matrix. There's some YouTube to show you how to calculate, to find eigenvalue, eigenvector for 2x2 matrix by hand. Okay, you have to do some determinant in order to reach that level. You probably can do 3x3 matrix to find eigenvalue, eigenvector, but for the higher dimension, we have to use computer. There are computational procedures to help you to find that. How can you find those values quickly and accurately? So that's a topic in numeric lineage algebra, and it's a field that people still working on. But use MATLAB or those languages, you can you don't worry about this. You just use it as a function. You get the values. Yeah, so that's what I talk about. So we do this called a decomposition. So you have A, you decompose it into eigenvector. So instead of transpose, this should be inverse. So inverse of the eigenvectors. And then multiplicate by eigenvalues, and then that again. So we will play with those using example in MATLAB. You can see, okay, I gave you a square matrix, then you found the eigenvector, eigenvalues, then you multiplicate them, you can recover your A matrix. So you do decomposition and the composition and come back to the original. So this procedure is called eigendecomposition. Okay. Now we jump to have a deeper understanding of this barcoding system. How can you label individual mRNAs? So this droplet capture cells and form this called a gel bead, and droplet, and in the center the gel bead, and covered with a 10x genomic barcode. Yeah, so that's what you load the cells and the gel bead in this row, and then partitioning OU in this row. I have never operated this, but a very standard procedure nowadays. By the way, this is not the only system you can use to do this single-cell RNA sequencing. This is a droplet-based. They are also the barcoding, multiplicating, barcoding-based. The past bioscience is also very interesting, slightly different technology. You have to have some quality control. You don't want to load a lot of died cells, and you want to have a staining and look at the ratio of live cells, and how under microscope you probably want to look at how dissociated cell look like, whether it's separated or some cell clamped together, and then in that case, one droplet may capture multiple cells. That's what we want to avoid. So we had this picture last week, and the green one is gel bead. The green, and your cell is in this gray circle, and the enzymes will break the cell membrane and let it release all the mRNA. Sometimes even the mitochondrial membrane has been broken and released into this droplet soap inside this bubble. As I said, the gel bead is covered with a 10x barcode. All the same, okay, for the same barcode, same bead, the same green barcode. So next droplet, you have a different barcode, so you know, okay, all the mRNA is from this cell, and all mRNA labeled with another barcode from another cell. So on each of this barcode, you also have another fragment. I still don't remember how long this UMI. This UMI is unique for all this oligo barcode. I think that's why the system and the reagents are so expensive, because you have to prepare this gel bead covered by the same barcode, and then on each of this barcode you have add another fragment. UMI is a unique label. Then you followed with a poly-T tail. So poly-T, there's a lot of TTTT, and they will tend to bind to the poly-A, because eukaryote gene expression, they have a mRNA normally have a poly-A tails, and so this part will bind to the poly-A tail, will enrich or capture those mRNA inside droplet. Okay? Is that clear? Computer science background people. Poly-A, poly-T. So mRNA, when you transcribe, it's always, a lot of them have a poly-A tail at the end. So you capture those poly-A tail, you basically found all the gene, protein coding genes transcriptions. Yeah?
So the gel beads only label one specific type of cell, or is the mixture of different cells?
It should be just one cell.
One.
Yeah.
So each specific cell type has gel beads.
It's not cell type specific. It's just label this individual cell. It doesn't matter which cell type it is. Yeah, it can be B cell, can be T cell, can be macrophage. No, because this gel bead only contains one cell, so there's no cell type of problem.
How is it decided how many
How many cells in there?
which type of gel beads are going to be used? Like, is it like a fixed number that's 10 of these?
Yeah, so that's why the system is so expensive. So they have a protocol, have this device to guarantee majority of the droplet contains only one cell, either empty or just one cell. And maybe less than 5% you have duplets or more than one cell captured.
So that means before you add gel beads, you need to make sure there's only one specific cell type to be or something.
No,
No?
no.
How?
So if you have a primary tissue, you have different cell types.
Yeah.
You just separate all the cells, let them to be independent. Yeah, separate.
Like sorting them out,
Yeah,
and then you add the gel bead to label.
yeah. The capture each of the cells with a droplet with one gel bead in there. Yeah. So this protocol sometimes is harder for the neurons. For example, this irregular shape and elongated axon in there. So they also have a solution of to extract nucleus and then so do a single nuclear RNA sequencing. Same idea, okay? Make them separable. So once you're labeled, so poly A tail and then this mRNA has been captured or fished out, and then you do this RT, reverse transcription, and to make this into ready for the sequencing library preparation. So UMI stands for unique molecular identifiers. Basically, just a barcoding. Okay. So the green 10x genomics 10x barcode tells you the gel bead, the cells. Well, the UMI tells you the fragment of this particular fragment. So at the end, you will put everything together and to sequence to get this information from a barcode, UMI, and a piece of mRNA from the gene the mRNA has been transcribed from. So in the middle, you have cDNA amplifications through the PCR reactions. And we want to have PCR reaction happen for each mRNA just once. But we have no control, so you may have a biased or amplification for particular mRNA, so make multiple copies. So UMI will be count at the end, just one copy, no matter how many copies you make for that UMI, you only count once to get rid of those cDNA amplification bias. Okay? So certainly we don't want to have just particular UMI labeled mRNA had been amplified too many times. We want to evenly cross all the mRNA molecule and to get one copy count for each of them. But at the end, we can use UMI to control for that amplification bias caused by the PCR reaction. Is that clear? So at the end, you will guarantee you count UMI uniquely just once, no matter how many copy of UMI you get from your end results. And also make sure the combination that's from that cell. Okay. So those are some library preparation make ready for Illumina short sequence. Basically, it will use pair ends, and you have designed the library so this end will read 10x barcode and UMI, and that end will read poly A captured some fragments from the gene transcripts. And because these two reads, it's pair end, so you know this label barcoding with that gene is linked. You can work out at the end through the pair end pairs ID. Again, so you need to understand 10x barcode is unique to each cell, and within each cell, UMI unique to each mRNA, cDNA transcription. So this is called R1. R1 contains these two barcodes, and R2 contains about 90 base pair of cDNA from mRNA. So using this 90 base pair, you can search on the genome of your reference and see, okay, this 90 base pair is from this gene. So you know the name of gene for this transcript. Okay, I hope on your mind you know how the unique cell and the unique mRNA can be labeled, and how the information of which gene this transcript is from can be whatever way you can trace it back. Is that okay, everything? Yeah. I hope you solved the problem on the class. There are some details of this, like this is 16 base pair and UMI is 12 base pair, and I remember they changed a little bit recently, maybe make a longer barcode. Then there are sample index, some base pair. If you want to go to Illumina sequence, there are another system, like how can you label those paired sequences. Yes? Mm-hmm. Mm-hmm. Yes, so I don't know, are you talking about the read 1, read 2, something like that? Oh, so how can we use UMI count? Yeah, I will show you examples later. There are some real examples later. Yeah. Yeah.
I have a question. What is that 1 5? I didn't understand that. Like the R2 portion is put up in the R2, like
Is on the slide or I wrote there?
That's the first one.
The first one, this one.
Yeah.
Oh, this is old procedure. They changed. They get rid of this. So index one file. I don't know. Yeah, it's redundant. Yeah, but now I don't think they have this anymore. Yeah. Now for each sample, you send your cells and out. You only have two files, R1, R2. So this old version, they have some index file in the third file. Yeah. That's FASTQ, yeah. Illumina FASTQ files with special labels, so you know. Yeah, I think that's R3 talking about. So they don't have this anymore. I don't remember. So I need to update my slide as well. Yeah. So again, this R1 cover this, so tell you cell identity and UMI, the non-unique mRNA count, and then this tell you which gene they're from. And because they are paired, so you know they're all everything linked. You don't need to pair because the cell ranger will recognize those pairs, will stretch them together.
I wanted to ask what does it mean by 12 mer? If we go back to the slide, it says
Mm-hmm.
the sample index is 6 base pair sequence and 10x barcode is a 16 base pair.
Yeah.
The UMI is a random 12 mer.
Yeah, the UMI.
Mer is, what does mer is?
Polymer, so yeah, so just base pair, 12 base pair random, yeah. So if you look at their technique documentation, that's their older version probably. So that's their 16 base pair, 10x cell barcode. You have a long list, the combinations. So from 1 to, there's like 700, more than 700. So they can prepare more than 700 with gel bead, each covered uniquely with those barcode. So you have a power to separate so many different cells. And for each of these, each barcode gel bead, they also covered with either 10 base pairs, the old version, or 12 base pair in verse 3, unique UMI. Okay, so each of these will come out and labeled again with all these UMIs. So each gel bead is one of these covered by all the UMIs and the poly T tails ready to capture all kinds of mRNAs in the droplet soap. Okay? So at the end you sequence, you get this, and you get this, and you know they're from different genes or same gene, different copy. So we send the samples to our genome center or whoever have this controller. They will run this. Normally, they will send you a link with a PDF download, and you can download those R1, R2 sequence file for each sample. You have 2 files. And you download and you put them into Cell Ranger. Cell Ranger, unfortunately, has no Windows version. It's rely on Linux operation system, and because their mapping or some other components in the pipeline require Linux machine only. So this pipeline will read those FASTQ files and make match and find out cells, find out genes by mapping the reads onto the reference genome. So you have to provide to the cell ranger. Number one is for each sample 2 files, and R1, R2. Number 2, tell the cell ranger what is the reference genome. The reference genome nowadays have a human and a mouse, and the cell ranger will find the genome sequence and also gene structure for human gene or for mouse gene, depends on your definition, your specification, and then try to map those rates onto those location and to tell, okay, this gene is from this cell, from that, this fragment, this UMI from which one. If you, by chance, you make mistake, say human sample, you gave a mouse reference, it still work, but not as good as a human because some of the human specific gene will not have any hit in the mouse genome, or mouse genome have multiple copies, so you get a slightly low quality readout, but still, but you have to know what sample you are working with. Some very basic QC or the quality control for the library will be able to generate and also have a quality report at the end. We will see an example. Then they require, you can use Looper browser and to visualize some of those outputs. If you want, you can use, but we don't have to. We are going to get the matrix, use MATLAB to do our thing, okay? But similar, okay? If you found this very easy to use, you can try to some of the basic class string should be okay. So I think this diagram shows some of this so-called aggregation. If you have multiple samples and some sample sequence deeper than others, in that case, people probably want to unify or normalize the sequence length in order to make them more comparable to each other. So in that case, this procedure, which also in the 10x genomics cell ranger, will subsample the rates. So it gave sample 1, say 5 million rates, and then sample 2, 7.5 million rates, and then the program will use the smallest amount of the sample to subsample your 7.5 million to 5 million to match the samples to get rid of a lot of information to make them comparable, and then run this and make a matrix, make them so-called comparable. But if you have like 5 samples, then there's only one sample sequence really low. This is not very good because it will sample to the lowest number of the samples to subsample, so you waste a lot of rates. There are options, and you can find. So in this is a barcode process, and the start. Start is a Linux-based aligner, so help you to align each individual read onto reference genome to find where the gene is, which gene the reads come from. And then the transcription count, just count how many copies of that particular gene expressed. Then you get the cell gene by cell matrix or cell by gene matrix, whatever. And that's downstream analysis. So back to Anastasia's question. So she got confused about the counting. Basically, how can you count without count multiple copy of a particular gene, not due to the upregulation of the gene transcription, but it's by PCR bias. You want to use UMI, you count only once if you have a multiple copy of the same UMI. So to control for that particular bias. I'll show you some examples. Again, we don't have this. We have R1, R2 arrays. That's a file name, and you have a sample name, and that's from Illumina sequence, and which learn and which batch. Okay. Then you have these two. You tell the program where your FASTQ file is located and where the reference is. Okay. So here is an example why the UMI help you to get rid of this PCR amplification bias. So cell 1, you have this particular cell barcode is indicated red. So they are all the same, they're from the same cell. And this blue cell is because it knows it's from blue cell, it's the same green cell and the teal cell. And your next is UMI. So this UMI and this UMI are the different ones labeled with different genes. So you count this gene for this cell, and UMI 1, so 1, 1, 1 for these three genes. Same here. For cell 3, and you found the same gene, but labeled with the same UMI. In this case, this fragment and this fragment is amplified not from the real mRNA one copy. It's from one copy, but multiplied by PCR twice. So we don't count this 2 times. We only count once by collapse those U. Should be only one gene because it's from one mRNA molecule in the cell. That's a unique particular copy.
Let's say certain gene is upregulated, so that means we have more
UMI, yes. So if you have a highly expressed gene in the cell, you have in the soup, you probably have thousands, hundreds of copies. In that case, you certainly will, by chance, have a more UMI label onto multiple real copy of that. Above that, you may have a amplification bias. This particular UMI got multiple copies, but you still count once. Yeah.
So in this case, does it fix the error that happens, or is it observation?
It's not an error, but it's unavoidable situation. PCR will amplify.
So how do we know if it's 2 reads that we should be counting as one molecule, or if it's upregulated gene because multiple copies are there?
You count UMI numbers. Yeah. An example like this. So this ARL1 gene have upregulated in cell C, cell 4. So by chance, you have 2 individual UMI and all labeled this gene. So because they are 2 unique, and the level is higher doubled. Yeah. rather than like this, just two UMI, but it collapse into one count. Two UMI is the same, so it's two copy of the same UMI, basically, the count one. Okay? So, yeah, so just remember, we use UMI as a count for individual mRNA molecule in the cell rather than the number of reads mapped to that particular gene. The purpose of the collapsed UMIs is to control for the PCR bias, amplification bias.
So we use coding to remove multiple instances of the same UMI
Yeah,
to differentiate between gene
Yeah.
upregulation
Yeah.
versus
Yeah.
et cetera.
yeah. You don't have to do it yourself because Cell Ranger, the pipeline, in this step already has already done this for you. So you just have to know conceptually and why we say UMI count rather than the mapped reads of the count. At the end, the Cell Ranger will generate this HTMI report to give you the sample estimate number of cells, and then there's some per cell average rate, median number of genes detected in each cell. You probably want to go through some of those percentage and then the shape of this. So each barcode is a droplet, okay? So you have over 700,000 barcode for the system, and then for each particular barcode, text barcode, we count how many UMI in there. If it's empty droplet, you will have no UMIs. So a lot of this barcode, the cells, so-called droplet, with non-UMIs. Some of this may have environmental mRNAs in there. So you start to see some of the UMIs with some of these empty droplets, but suddenly you have a droplet which capture the cells. Then you start to see, okay, UMI count for this particular droplet start to increase. And at the top, you may have duplets or multiple cells in one droplet. In that case, you have really high UMI numbers in those. Either the cell is bigger or highly transcribed. But you look at the shape of the curve, you may have some feeling about the distribution of your empty droplets, your environmental noise mRNAs, duplicates, triplicates, and the cells with a lot of transcription. Or in the middle, those are the regular single cell UMI counts. Okay?
So, like we did one of the runs and we received like number of cells were 35,000.
Mm-hmm.
But what should we do then to enhance the number of cells that we receive?
I never see 35,000. This is out of the range of the 10x genomics. This number is a little bit too high for me. So normally 11,000 or 10,000, 8,000 is all regular range.
Because we recently received this report and it said that the cell number received was around, like the cell number counted without filtering was 35,000, about 35,000.
I think the way they decide this is by cut the curve. See, if I accept this cutoff, then I will have 10,000 cells. If I relax my cutoff a little bit, and then we have certainly quite a lot. So it depends on how many UMI count we will accept as a droplet capture one cell. There's no good way, and you just compare to other experiment system, do multiple, and then compare those curves. Maybe you have broken cells, and so in the cell preparation, there are a lot of environment mRNA, so capture those. There are a lot of contaminations. The pipeline still consider there are enough UMIs in there as a cell, but in reality, maybe because your cell has been broken, and then their environment mRNA somewhere that make this false calling of cells. Yeah.
Can you set a parameter as a cutoff value?
Yeah, I think so. If you dig into the pipeline, now they open sourced, so you can change. Otherwise, by default, it would give you the curve, and by default, they have two versions: is before filtered and after filtered. So you select. So normally we use a filtered processed matrix. Yeah.
The higher number was without filtering.
Without filtering, it's not reliable. Yeah. Okay.